Hi guys, so in this video we're going to talk about epistasis. Epistasis is another type of inheritance, and it's defined as when the allele of one gene affects or masks the expression of another in the phenotype. In simple words, this means that the expression of one gene depends on the other, or if you like, one gene can interfere with the expression of another gene. But to understand this better, we have to give some examples. So let's look at mice. Now, mice have genes that control their coat color, or if you like, their fur color. We are going to look at two genes. Always in epistasis, we are looking at two genes, or if you like, we are looking at two different traits. Therefore, we always do dihybrid crosses. So let's take one gene, let's call it gene A. Well, think that gene A is responsible for the production of melanin. Now, melanin, it's a pigment, or if you like, melanin, it's a color, and it's usually the black color. So if there is no melanin production, then it means that the mice will be white. Now, this gene A has two alleles, A and small a. Now, the dominant A leads to melanin, so it will produce melanin, and the recessive allele will not produce any melanin, so no melanin. Now, if you are homozygous recessive, it means that you're not going to have any melanin, so the mouse will be white, will have a white coat. And when you are homozygous dominant or heterozygous, it means that you are going to have melanin. Now imagine that we also have a second gene. Now let's call it gene B. Now gene B controls distribution of melanin. Now this gene has also two alleles, dominant B and recessive B. The recessive B will form a uniform black hair, which means that the mice will be black. Now the dominant B will express for black bands. This means that the black color will be spread out but not evenly, so, or if you like, is not going to express that much. So basically it's going to form something like a brown or gray hair for the mice. Now, as we explained before, epistasis is when one gene interferes with the expression of another. So take this example here. If there is no melanin, so if a mouse is homozygous recessive, then it will not make any melanin, so gene B will not actually do anything because there is no melanin to distribute. So gene A will affect the expression of gene B. So if a mouse has this genotype, then you will not see the effect of gene B on the phenotype because there is no melanin to distribute. But if a mouse is homozygous dominant or heterozygous, then distribution of melanin will be seen in the phenotype and either will have black bands or uniform black hair. Now, let me rewrite those symbols underneath and let's try to do some examples, and hopefully you will understand and get it quickly. So these are our symbols. Let me give myself a little bit of space. So let's start with a pure breeding cross. So let's say that the parents have this genotype A, B, B, and the other parent it's A, B, B. Now the gametes here will be A, B, the gametes here will be A, B, so these are the gametes. So the F1 generation is going to have this genotype A, B, B. What this means? That melanin is going to get produced and they are going to have black bands. In other words, if you see a mouse with this genotype, then the mouse will be brown or gray. And we call this mouse a goody. A goody mice are the ones that are actually brown. Now, if I take the F1 generation and cross it, so let's say F1 and I take two parents that are actually heterozygous for both genes. So things will get more complicated and we're going to have more phenotypes. So gametes can be A, B, B, A, B, B. So we carry out a normal dihybrid cross. We make our Punnett square. So 4 gametes for each parent, A, B, A, B, A, B, A, B, A, B. So hopefully you've learned by now how we carry out these crosses. We make our lines and then we do our genotypes. So these are all the genotypes we're going to get from this cross. So the easiest ones to start with is the ones that don't really have melanin. Because if gene A is not expressed, then gene B is not going to express either. So let's find the ones that are homozygous recessive for gene A. So we have one here, one here, one here, and one here. So we have these mice here that will not express. Okay, so let me write here white. We have 4. So these mice here that are not expressing melanin will be white. They are going to have a white fur or a white coat. So all the other 14 mice will express melanin. So gene A is going to express, and since gene A will get expressed, it means gene B will determine how melanin will get distributed. So I will use a gray color to show the ones that are actually black. So I'm going to write here black. Let's find, to identify the black ones, we have to look for the ones that are homozygous recessive for the B gene. So we have one here, one here, and one here. So we have 3 mice that will have black coat. Now the rest, 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 mice, let me take a brown color. Now 9 mice will have the agouti phenotype. Agouti is for brown color. So if you see now, if we cross 2 heterozygous parents for both genes, then the ratio we get is 9 to 4 to 3. So we get 3 phenotypes in total. Okay, so you can see here like melanin is present and it forms black bands. Black bands means brown. So the same here, here, and here, and we have 4 ones that will be albino because gene A is not expressed, so no melanin will be there, so definitely they will be albino. And we have 3 other genotypes that will have melanin, but melanin will be distributed evenly around the coat of the mice, so all 3 mice will be black. So now let's see a second example of epistasis. Epistasis will not, of course, only affect this kind of reactions, but also involves in sequence of reactions or biochemical pathways, if you like, where enzymes are involved. And let's give an example of that. So imagine that we have a starting molecule which starts the biochemical pathway, and this molecule here can be converted through enzymes into a red molecule, and that red molecule can be converted into a green one. Imagine that this takes place in leaves. So a leaf of a plant will be green if enzyme A, which is responsible for the first reaction, catalyze the reaction, and then to go from red to green we need a second enzyme, which is enzyme B. So imagine that this enzyme A is controlled by a gene A, and this enzyme B is expressed by a gene B. So in this case, imagine that dominant A it's for the functional type of the enzyme, and the recessive one, it's for no enzyme at all, or if you like, a non-functional enzyme. The same applies for B. So B, we make a functional enzyme, and recessive B, we make no enzyme or not a functional enzyme. So if this one here, if this specific leaf has this genotype or this genotype, it means that this reaction will take place and the starting molecule will be converted into red one. Now, if this same leaf has this genotype, homozygous dominant or heterozygous, it means that this reaction will also take place and the color of the leaf will be green. But if this enzyme is not expressed, so if we imagine that now our genotype is not this, but is this, then we're not going to form a functional enzyme, so this reaction will not take place, so the leaf will have a red color. So you can see that epistasis can also affect biochemical reactions or biochemical pathways. Therefore, this here is an example of epistasis because gene A can mask the expression of gene B. So if the enzyme A is not present, therefore the starting molecule will not get converted into the red molecule, so the enzyme B will not function at all. So enzyme B, okay, so the function of enzyme B depends on the function of enzyme A, or in other words, gene B depends on gene A. So this is an example of epistasis.